Glycolysis gave us pyruvate, but it only squeezed a tiny fraction of the energy out of the glucose. The real jackpot is still locked inside. So where does the cell actually cash in? Right here, in a cycle so central to metabolism that it's been called the hub of the cell. It has three names you will see used interchangeably: the citric acid cycle, the Krebs cycle, and after Sir Hans Krebs, who worked it out in 1937, the TCA cycle, short for tricarboxylic acid. And it's not just an energy machine. This cycle is where the breakdown of carbohydrates, fats, and protein all converge. Every fuel your body burns end up flowing through the same 8 reactions. So in this video, we will do it properly, all 8 steps, and at every single step, we'll look at the actual structure, which carbon loses its carbon dioxide, where the double bonds form, how oxaloacetate is regenerated, and by the end, it won't be a cycle you memorized, it will be a logic you understand. So let's begin. Where does TCA cycle happen? Unlike glycolysis, which runs in the cytoplasm, the citric acid cycle takes place inside the mitochondrial matrix, the innermost compartment of the mitochondria. And though no oxygen is directly used in the 8 steps, the cycle is strictly aerobic. It only turns when oxygen is available downstream to accept the electrons it produces. No oxygen, no cycle. Before the cycle can start, the pyruvate from glycolysis must be converted into cycle's actual fuel. This is called the link reaction or oxidative decarboxylation of pyruvate, carried out by the pyruvate dehydrogenase complex. One carbon is stripped off as carbon dioxide and NADH is generated, and the remaining 2 carbon acetyl group is attached to a coenzyme A to form acetyl coenzyme A. That acetyl coenzyme A is the molecule that actually enters the cycle. Fats and many amino acids also break down into the same acetyl coenzyme A, which is why it is the great metabolic crossroads. Here's the elegant design. A 2-carbon acetyl group joins a 4-carbon acceptor, oxaloacetate, to make a 6-carbon citrate. Then as the cycle turns, 2 carbons are released as carbon dioxide, and through a series of oxidation, the molecules is whittled back down to the 4-carbon oxaloacetate, ready to grab another acetyl group and go again. It is a catalytic cycle. Oxaloacetate is consumed at the start and regenerated at the end. So a tiny amount can process endlessly acetyl coenzyme A. The energy is captured mostly as reduced electrons carrier, 3 NADH and 1 FADH₂, plus 1 GTP by substrate level phosphorylation, releasing 2 carbon dioxide. Those NADH and FADH₂ then carry their electrons to electron transport chain, where the real ATP bulk is made. Now let's talk about the structures. Step 1. Condensation. Acetyl coenzyme A plus oxaloacetate gives citrate. The structure going in. 2 molecules meet here. Acetyl coenzyme A, CH3 C double bond O S coenzyme A. Focus on the 2-carbon acetyl group. A methyl CH3 and a carbonyl carbon join to coenzyme A through a high energy thioester bond. That thioester is energy rich. Its energy will drive this reaction. Oxaloacetate. C double bond O, COO minus, CH2, COO minus. This is 4-carbon acceptor, a keto acid with reactive ketone carbon that acetyl group will attack. The enzyme citrate synthase joins the 2-carbon acetyl group of acetyl coenzyme A onto the ketone carbon of oxaloacetate. A carbon-carbon bond forms. A molecule of water is added, the high-energy thioester bond is broken, and coenzyme A is released as free coenzyme A SH. The energy of that thioester bond is what makes this condensation strongly favorable. It is essentially irreversible. The structure we produce is citrate. Look at it. A 6-carbon molecule with 3 carboxylate groups. That's why the whole pathway is named the tricarboxylic acid cycle. Citrate is the tricarboxylic acid. Note the central carbon now bears a hydroxyl and it is a tertiary alcohol, meaning it has no hydrogen on the carbon. which turns out to be the problem. The cell must solve this in very next step. This is the key regulatory step and effectively irreversible. Investment of carbon, we have gone from 4 carbon acceptor to 6 carbon citrate. Step 2, isomerization. Citrate to isocitrate via cis-Aconitase. Why this step exists? It is a structural problem. Citrate is a tertiary alcohol. The OH is on the carbon with no hydrogen attached. You cannot oxidize a tertiary alcohol easily. But the next stage of the cycle needs to oxidize this molecule. So the cell first has to move to hydroxyl group to a carbon where it can be oxidized, converting the tertiary alcohol into a secondary alcohol. That's the entire purpose of step 2. Relocate the OH. Step 2a, dehydration. Aconitase first removes a molecule of water from citrate, creating a carbon-carbon double bond. The intermediate cis-Aconitase has the structure you can see in the notes. This is a transient enzyme bound intermediate. Step 2b, hydration. Addition of water. Then Aconitase adds a molecule of water back, but onto the adjacent carbon this time. The net effect is the hydroxyl group has effectively moved to the neighboring carbon. The structure we produce is isocitrate. Compare it to citrate. Same atoms, same formula. It is just an isomer. Hence isocitrate. But now the OH sits on the carbon that also carries a hydrogen. It is a secondary alcohol and that can be oxidized. Problem solved. Step 3, oxidative decarboxylation. Isocitrate to α-Ketoglutarate. The structure going in is isocitrate with its newly repositioned oxidizable secondary hydroxyl. This is the cycle's first oxidative decarboxylation, and the first carbon dioxide and the first NADH are produced here. Two things happen at once, an oxidation and a decarboxylation. The enzyme isocitrate dehydrogenase does these two jobs. Oxidation, it oxidizes the secondary hydroxyl group. The electrons and a hydrogen removed are handed to NAD+, producing the first NADH. And second is decarboxylation. It strips off one of the carboxylate group released as the first molecules of carbon dioxide. The structure we produce is α-Ketoglutarate. We have dropped from 6 carbon molecule to a 5 carbon one because one carbon left as carbon dioxide. And note that new ketone where the hydroxyl used to be, that carbon has been oxidized. It's now an alpha keto acid with a keto group alpha to carboxylate. Isocitrate dehydrogenase is one of the major control point of the cycle, activated when the cell needs energy, inhibited by ATP and NADH when it does not. Step 4, oxidative decarboxylation. Alpha-Ketoglutarate to Succinyl coenzyme A. The structure going in is alpha-Ketoglutarate. Our 5 carbon keto acid. This is the second oxidative decarboxylation. The second carbon dioxide and second NADH. And it's mechanistically almost identical to the link reaction that made acetyl coenzyme A. The enzyme alpha-Ketoglutarate dehydrogenase, a large multi-enzyme complex closely related to pyruvate dehydrogenase, does these three things: decarboxylation, oxidation, and attachment of coenzyme A. Decarboxylation removes another carboxylate as the second carbon dioxide. Oxidation, the electrons reduce NAD+ to second NADH. Third, attachment of coenzyme A. A new high energy thioester bond forms within coenzyme A SH. The structure we produce is Succinyl-coenzyme A. We have dropped again from 5 carbon to 4 carbon. And critically, note that high energy thioester bond at one end, just like in acetyl coenzyme A. This bond is energy rich and the cell is about to crash it in for a molecule of GTP in the next step. Pause on the carbon accounting. 2 carbon dioxide molecule have now left the cycle. We are back to a 4 carbon compound. The cycle has released as much carbon as acetyl group brought in. Everything from here is about regenerating oxalate and harvesting the remaining energy. Step 5. Substrate level phosphorylation. from succinyl coenzyme A to succinate. The structure going in is succinyl coenzyme A, carrying that high energy thioester bond we just built. The enzyme succinyl coenzyme A synthase, also called succinate thiokinase, breaks the high energy thioester bond and uses the energy released to attach an inorganic phosphate to GDP, forming the GTP. Coenzyme A is released as free coenzyme A SH. This is called the substrate level phosphorylation. Energy transferred directly from a high energy substrate to form GTP, exactly like ATP making steps in glycolysis, and needing no electron transport chain. Note, GTP is energetically equivalent to ATP. The cell readily converts one to the other. So many textbooks simply write 1 ATP here. The structure we produce is succinate. Look at its molecule. It's beautifully symmetrical. A 4-carbon chain with carboxylate at each end and 2 plain CH2 group in the middle. The coenzyme A and extra energy are gone. What remains is a simple symmetric 4-carbon diacid ready to be oxidized. Step 6. Dehydrogenation. From succinate to fumarate. The structure going in is succinate. Our symmetric 4-carbon diacid. Focus on the 2 central CH2 group. We are going to remove a hydrogen from each, creating a double bond between them. The enzyme succinate dehydrogenase removes 2 hydrogen atoms from 2 central carbons, forming a carbon-carbon double bond. These electrons are not energetic enough to reduce NAD+, so they are captured by FAD instead, producing FADH₂. Succinate dehydrogenase is the only TCA cycle enzyme not floating free in the matrix. It's embedded in the inner mitochondrial membrane, and it is complex 2 of the electron transport chain. So this enzyme physically links the cycle directly to ETC. It feeds its FADH₂ electrons straight into the chain. That's also why FAD is used here rather than NAD+. The structure we produce is fumarate. Note the new trans double bond between the 2 central carbons. It is specifically the trans isomer fumarate. The enzyme is stereospecific and makes only this form, not the cis isomer. Step 7. Hydration. From fumarate to malate. The structure going in is fumarate with a carbon-carbon double bond. We are going to add water across it. The enzyme fumarase adds a molecule of water across the double bond, a hydration reaction. One carbon of the double bond gets a hydroxyl group. The other gets a hydrogen. The structure we produce is malate. The double bond is gone. One of the central carbon now carries a hydroxyl group, a secondary alcohol, and the other is plain CH2. Fumarase, like succinate dehydrogenase, is stereospecific. It produces only L-malate. Why install that OH? Because the final steps need something to oxidize, and you are setting up the regeneration of oxaloacetate. Step 8. Dehydrogenation. From malate to oxaloacetate. The structure going in is malate with the secondary hydroxyl we just installed. This is the final oxidation, producing the third and final NADH, and it closes the loop by regenerating oxaloacetate. The enzyme malate dehydrogenase oxidizes the hydroxyl group of malate into a ketone, and the removed electrons and hydrogen reduced NAD+ to a third NADH. The structure we produce is oxaloacetate. Looks familiar? It should. This is exactly the molecule we started with the step 1. The hydroxyl of malate has become a ketone, regenerating the 4-carbon acceptor. The hydroxyl is oxidized to a keto group, and oxaloacetate is ready to condense with a fresh acetyl coenzyme A and turn the cycle again. Now the cycle is complete. Oxaloacetate accepts the next acetyl coenzyme A, and the whole wheel turns once more. This is what makes it catalytic. The oxaloacetate is never used up. It simply cycles endlessly, progressing acetyl group after acetyl group. Now let's quickly do the energy accounting. So per single turn of the cycle, which is 1 acetyl coenzyme A, step 3, isocitrate dehydrogenase, 1 NADH plus 1 carbon dioxide. Step 4, alpha-ketoglutarate dehydrogenase, 1 NADH plus 1 carbon dioxide. Step 5. Succinyl coenzyme A synthase, 1 GTP. Step 6. Succinate dehydrogenase, 1 FADH₂. Step 8. Malate dehydrogenase, 1 NADH. So per turn we got 3 NADH, 1 FADH₂, 1 GTP and 2 carbon dioxide. And here's why those NADH and FADH₂ matter so much. Each is an electron carrier that will deliver its electron to the electron transport chain, where oxidative phosphorylation converts them into the bulk of the cell's ATP, roughly 2.5 ATP per NADH and 1.5 ATP per FADH₂. The citric acid cycle itself makes only 1 GTP per turn directly, but the electron carrier it produces are where the real energy pays off is stored, ready to be unlocked in the final stage of the respiration. Now let's quickly recap. 8 steps starting from acetyl coenzyme A. Step 1. Condensation. Acetyl coenzyme A plus oxaloacetate gives citrate by citrate synthase. Step 2. Isomerization. Citrate to isocitrate via cis-Aconitase is done by Aconitase. Step 3. Oxidative decarboxylation from isocitrate to alpha-Ketoglutarate by Isocitrate dehydrogenase. Step 4. Oxidative decarboxylation from alpha-Ketoglutarate to succinyl coenzyme A by alpha-Ketoglutarate dehydrogenase. Step 5. Substrate level phosphorylation succinyl coenzyme A to succinate by succinyl coenzyme A synthase. Step 6. Dehydrogenation from succinate to fumarate by succinate dehydrogenase. Step 7. Hydration from fumarate to malate by fumarase. And last, step 8. Dehydrogenation from malate to oxaloacetate by malate dehydrogenase. Per turn we got 3 NADH, 1 FADH₂, 1 GTP and 2 carbon dioxide. For per glucose, just double it. If the logic of Krebs cycle now makes sense, why the OH has to move, why 2 carbon leaves, why FAD shows up at exactly 1 step, that's what bioarchive is for. That's it for this video. Keep learning, keep achieving. I'll see you in the next one. Thank you.